Okay, again, thank you all for joining us. We have had more come into the webinar, so we're going to get started. This is on INGA. I named these after my grandma and her sisters. Emma is the one we do for environmental mold and mycotoxin assay that you've heard of with our environmental test. INGA is her sister, but it really stands for Integrated Nutritional Genetic Analysis. We are learning that we need to date our webinars because so many people don't do this on the web, and you don't know whether it's something recent or something that was done 20 years ago. So we are saying this was presented August 6th. It's presented by Real Time Labs. My name is Dennis Hooper, and I'm a consultant. Again, we don't talk about treatment plans. We don't see patients here. And my associate, Shalini Murladhar, is going to be here today to be introduced into these webinars. So you'll hear her. We tried to get her picture in, but my computer wouldn't allow it. So it has to be that it's her picture in my computer. So you'll see a picture of her next time with a real picture of me as I'm getting older. Okay, what are the goals of our webinar? Today we're going to learn what MTOR is. We're going to learn what its relationship of all the genetic variants are to the environmental exposures. We're also going to learn how to explain some of these relationships to our environmental customers, but we're going to learn that in layman's terms. And we're going to learn how is mTOR related to pesticides, environmental toxins, such as mycotoxins, bacterial toxins, and Lyme disease. Now, this is a very complicated subject, so we've had to distill this down so that you can use it realistically to work with your patients as well as your clients in understanding how the environment affects us in our environmental exposures and how it affects our genetic analysis. So bear with us. This is going to be a series of 5 webinars that we call Dr. M instead of Muraldhar because it's difficult for me to say. So we call her Dr. M, and she will be talking today a little bit as well. The first thing we're going to do is show you a 6-minute.
Life runs according to a blueprint, which is the DNA. So we can refer to the DNA as life's own sequence. Now, what is DNA? First, we all have proteins and enzymes in our body. These enzymes and proteins are created from different processes, all of which are coordinated by the DNA. DNA is the reason why one person is different from another. Now that we understand what DNA is, we can talk about the gene. Your gene is made from the DNA in the form of codes. These codes usually take the form of a 4-letter combination containing alphabets of chemical basis, i.e., the ATGC, which is also called the nucleotides. If the cell is ready to create any protein molecule, it takes instruction from the gene sequence. Thus, the gene sequence determines the type of protein molecule that will be created, and therefore, the proteins are unique eventually, especially in shapes and functions. The uniqueness means they can control various segments of metabolism, cellular anatomy, and physiology. Another important component to discuss is the genome. In simple words, all the genes found in the body of any living thing is collectively referred to as a genome. For humans, our genome comprises about 20,000 genes. Genome, however, differs in composition. another reason for our uniqueness as humans. And this leads to differences in gene sequence, which ultimately results in variations in function, structure, and the amount of protein produced. Now, let us compare mutation and polymorphism. Mutation is the consequence of rare differences in the gene sequence. When this happens, its effect on the population is negligible, and, most times, we can predict the outcome. Polymorphism, on the other hand, is more rampant, but with less serious effects relative to mutation. Also, it is usually influenced by some environmental factors. You can have polymorphism in different forms, but we will be discussing only the most common type today, which is the single nucleotide polymorphism, or SNP. SNP simply means the swapping of one nucleotide for another. Note that the genome contains up to 10 million SNPs. You will find people use SNP and mutation interchangeably. However, an important point about SNPs is that they rarely affect the protein created, and in those rare cases, they are not of medical importance. One of the most notable effects of SNPs is the distortion in the nutritional requirements. This causes the body to respond to most lifestyle and dietary factors differently, and this is where genetic testing comes in. With genetic testing, it is possible to get vital clues that could help in coming up with a bespoke special diet and protocols for each person. Although nutritional genomics as a field has continued to evolve, there is still more to do as per the exploration of SNPs. In-depth investigation of these phenomena will reveal the beneficial forms and how they can be applied clinically to the interpretation of the genetic test information. A couple of factors influence the final interpretation of any SNP on the psychology of an individual. These factors usually include the diet, environment, as well as the epigenetic and genetic factors. The field of genomics, if explored well, is capable of making common SNPs very relevant clinically, especially when it comes to nutrition. From our discussion so far, it is clear that the DNA has so much to do in the future of human health and nutrition. Let us start with the Integrative Nutritional Genetic Analysis, INGA Test, which was inspired by the need to understand DNA with regards to human health and nutrition. With the INGA test, it is possible to find out the most beneficial enzymes in your body and how to best use them to improve your health. By identifying the different SNPs in your gene, all dietary inconsistencies, if any, will be revealed. Also, if you have an insufficient amount of nutrients or antioxidants or none at all, this test will also make this apparent. The dietary inconsistencies, if any, can be easily solved by creating a personalized nutrition protocol for the patient based on their genetics. With this protocol, the patient gets vital supplements and nutrients that can make up for losses and neutralize the inconsistencies. They also help to ensure the judicious use of vital enzymes, as well as the creation and support of healthy cells. If need be, the patient may also undergo detoxification therapies. This is not as complex as it sounds, especially when you consider that our well-trained nutritional consultants will be with you at every step of the way. And by towing the path of nutritional support, we will arrive at a viable maintenance protocol in the end, while providing lasting solutions to all issues. The patient also gets a bespoke maintenance program that meets all their nutritional health requirements. The more we dive into this progressive approach of providing excellent help and support to genetic variants, the more we come up with excellent solutions and answers to several challenges. So, why not join us today?
Okay, I'm sorry that we had a little bit of a problem in connections. You can reach this video again through our website, www.realtimelab.com. The reason that I wanted to play that is so that you understand the basis for why we believe that MTOR is very important in how the environment affects our lives. So mTOR stands for Mechanistic Target of Rapamycin. I know it just rolls right off your tongue. mTOR is much easier to remember. This product, or mTOR, coordinates mammalian cell growth. In other words, human cell, animal cell growth, and the metabolism with environmental inputs. These include nutrients and growth factors, and also inhibitory substances that we're going to go through. Again, this seminar, or this webinar, is only about 20 minutes, 25 minutes long today. And each one is, we want to keep them around that thing, that time limit, so that you don't lose interest and that you take something away. So the central role of mTOR in regulating our functions are many fundamental cell processes that associate with protein synthesis to actually the cell dying, and this is called autophagy. Anytime during this seminar or webinar, you can email your questions in, and we will be glad to work with them either as you're going on that slide or as at the end. mTOR is also deregulated so that signaling coming out of mTOR can be implicated and implemented in the progression of cancer and diabetes. And these are not things that we're saying, but we have reviewed the literature, and we've seen that mTOR, if it's affected, affects not only how we progress in cancer and diabetes, but also in aging. The most important paper that you might want to have available and read about is from Robert Saxton and David Sabatini, that was written in the Journal of Cell in March 9, 2017. Saxton and Sabatini are from MIT and other institutions, and they are very reputable, and it's a paper that you should have in your over the next few weeks.
Thank you, Dr. Hooper. This chart may look a little hazy, but there's a reason why we have it color coded so it's easy to follow. Like, you can look at it that mTOR is the center of the lifestyle and the environmental factors and the genetic SNPs that influence this pathway. mTOR and autophagy are opposite to each other. Whenever mTOR is regulated, autophagy is inhibited. Like Dr. Hooper said, autophagy is something that clears pathogens and cell debris after cell death out of your body. So whenever mTOR is regulated, autophagy is inhibited, and this can go on as a loop in the presence of tumor suppressor genes, rapamycin, etc. So mTOR is active, autophagy is inactive, and that can be turned on by these factors, and then again regulate mTOR. So if you look at this chart, the very top yellow coded boxes are all the epigenetic factors that influence the mTOR signaling. For example, your dairy intake, your proteins, plastics, electromagnetic signals, your telephones, sugars, foods that you consume, B12, vitamins, pesticides, etc. These are all the external epigenetic factors that influence your mTOR. What genes do they influence are all coded in pink. So these are the SNPs that are studied or revealed in your INGA test to see whether these genes are variant, whether it's a homozygous variation or a heterozygous variation. If you have variations in these genes, then it would further influence some of the factors that are involved in the mTOR signaling or the autophagy. For example, PON1 is a pesticide gene. Any variants in this PON1 gene or a SNP will result in able to detox yourself from any external pesticides that you might have intaked, which influences your glutamate levels in the body and affects the mTOR. The innermost circle is a green colors are all the hormones and amino acids that are the factors that may influence the mTOR because they keep the mTOR regulated. Once the mTOR is regulated, it affects your cell functions anywhere from mRNA translation, lipid synthesis, or nucleotide synthesis at a cellular level. So you can see that how mTOR regulates, but is influenced by many factors, from environmental factors to genetic SNPs and various pathways that are involved in this process. There are many oncogenes as well that influence mTOR, and the SNPs that are associated in this studies are MRF2, SOD, and catalase enzymes. So basically, mTOR, when regulated, it plays a very important role or a fundamental role in cellular processing, such as protein synthesis, lipid synthesis, nucleotide synthesis, etc. When it's deregulated, it could lead to cancer, diabetes, and aging. We will talk more about these slides as we go in the depth. So this whole pathway is simplified in our INGA test in a form of a pyramid. So you can see the pyramid is color coded as well, from the bottom being green, white to yellow at the top. The goal of this SNP INGA testing is to see if you have any variants in genes that influence your copper, iron, magnesium, your hydrogen peroxide cleansing, etc. And the top of the pyramid that is affected is the B12 and the folate levels as well. The goal of the INGA test is to look at the bottom three green boxes or columns or rows to see what are the genes that are influencing in your detox. The reason we would like to go from bottom to the top is because anything changes or variants you have will have a snowball effect till the top of the pyramid. So it is important that you focus and pay attention to your lower end of the pyramid to resolve and see how you can support through supplementation or any nutritional protocols to support these losses, which eventually prevent a loss at the very top of the pyramid. Patients come back with only information such as MTHFR being issue, but the goal actually should be to look at the lower levels, how those environmental toxins or electricity sensitivity, etc., are affected, which may be the result for your B12 and your MTHFR folate. So INGA test focuses on all of these pathways, and the INGA test, once it's completed, it's a saliva test. We send you the kit, you collect the saliva, and we send it back for the analysis. It generates a 68 pages to 70 pages report. I know it's a lot of information, but definitely necessary to understand what really is going on at the level of metabolism and detoxification and other effects on your pathways. So in the future, we will future webinars, we will discuss some of these pathways and tell you what variants or what gene variants are important for each of these blocks that may have an long-term effects in patients. Dr. Hooper?
Thank you, Dr. M. Yeah, the entire reason for this is we get a number of calls saying, well, our nutritionists or our environmental testers have said, we need to have this test done. Well, we don't understand why, because our doctors have told us our MTHFR is a variant, and therefore that will never get better. And I have to say, we're going to have a seminar or a webinar just on MTHFR and C4A, because those the literature does not point to having an issue of MTHFR and C4A as a problem with these environmental toxins. We have not seen where the MTHFR is an issue for in the most, we're trying to be as respectful as we can to those who use it. And we will cite that literature in the future. So here's the history of MTOR. In 1964, a Canadian expedition went to the South Pacific. What a great assignment. And they went to an island called Rapa, which is in the Easter Islands. And they were told to find some soil to get an antimicrobial agent. Well, they found one in bacteria, and the bacteria had all this antifungal, immunosuppressive, and anti-tumor properties, and it was all published in the later years. And they named this compound rapamycin from that island of Rapa. And now, 20 years later, they believe that mTOR protein has an enzyme which works in the human and animal cells that signal cell growth, and they're associated with the environmental conditions that we're going to talk about in the future. But mTOR plays a very fundamental role in all the cells and all the organisms and their physiology. In other words, how they're growing. mTOR is a protein kinase. That's an enzyme that brings together all these other subunits of proteins. And in order for the cells to grow, they have to produce proteins and lipids and DNAs and everything. And so they're associated, as Dr. M pointed out, in all these protein synthesis, the glucose, the protein turnovers, and how they respond to the environmental stresses are compatible or incompatible with growth. So your involvement at the environmental level is extremely important to these patients. You may not think so, or they may not think so, but you need to emphasize that all the things you're finding associate with their cell metabolism. So the perspectives that we're going to go forward with in mTOR shows that it plays a central role, you saw that, in sensing all these environmental conditions. It regulates all the aspects of metabolism at the cellular and the organism level. And it has mechanisms that have been characterized in all different areas of medicine. And we're going to be concentrating on future work that these sites are doing, as well as what Real-Time Lab is doing in the forthcoming workshops and webinars. When we do this, we are going to be offering CMEs, if we do workshops in the cities that we are traveling to, we will be offering continuing medical education through the University of Nevada School of Medicine. And these are for the MDs present and for the medical people present. These are very important, as you know, for your licensing effect. I would caution, and I put caution in high capital letters, and I put bolding, that there are some places that say that there are CMAs given. And when you need to look and see, where are these CMEs coming from? As we have found that they're from home universities that aren't accredited and are not able to be recognized by the boards in their states that give the medical licenses, etc. And they're just not feasible. So you're wasting your money. You may be interested in listening, but you can't get CMEs that are going to be recognized. So we don't do that at Real Time Lab. We're being straightforward and honest with you. So the future work that we're going to discuss will focus on the development of the MTOR targeting therapeutics. And what is the MTOR going to look at in your area, if it's environmental or if it's in therapeutics? We are going to understand at the environmental level what MTOR does and how the environmentalists can help curb the effects of the environment on MTOR pathways. The next webinar is going to be specifics of MTOR on Lyme disease, and that's going to be August 20th, 2019 at 11 a.m. And next time we'll have it more together as far as our video. We won't be showing another video. But again, we're going to show how Lyme disease is associated with MTOR, how the mycotoxins are related, and how the MVOCs are related to MTOR, and other things in the environment. So we have some questions, or we are anticipating, and maybe not, but the question that we have, first of all, is how do environmental inspectors use this with their customers? And we think the best thing to do is discuss that the mycotoxins, the bacteria, and the fungi that you collect affect all of their cells. They all will come to you and say, oh man, I feel sick. I've never been getting any better. I've gone to 20 doctors. Nobody's helped me. And why should I come to you at all as an environmentalist? What are you going to do for me other than tell me that there's a bunch of fungus or something there? Well, the fungus itself and the molds aren't going to be as helpful as if the mycotoxins or the bacterial toxins are found. So we're going to be working with the inspectors on looking at the MVOCs as well as the mycotoxins. So those questions on how do you, we're going to work with you on those questions. If you have questions that your people bring up, let me know, and we'll put them on our next webinar, as well as we'll put them on our website. You can email your questions to me at Dennis, DENNIS.Hooper, HOOPER, at realtimelab, REALTIMELAB.com. We hope this is going to be informative, but it's the newest techniques, newest thoughts on why environment is affecting patients and how you can help them as environmental inspectors. So with that, we will stop this webinar, and we anticipate seeing you again on August 20th. Thank you.